Okay, we're going to talk about chapter 3, which is the cell. The cell is the smallest intact unit of life. It's the function unit of life. All living things are made up of cells. Going to humans, we have about 70 to 100 trillion cells in our body. Now that is the standard human, 5'8", 70 kilograms. Estimated around 70 to 100 trillion. Most would say around 100 trillion. There, with many classifications, we have 210 different cell types. 210 different cell types. So 100 trillion cells, but 210 different cell types. This process of differentiation. We have two broad groups. We separate the somatic cells, which are all the cells in the body, from the gametes. The gametes are the sperm and egg. The somatic cells have twice as much genetic material as your sperm and egg, because the sperm meets the egg and restores the genetic material amount. You can read on some of the things that historically, as far as cells coming about, and this, I'm not going to get into that. We'll say that there are two basic kinds of cells, two broad divisions, the prokaryotic and the eukaryotic. Karyon, K-A-R-Y, is an old name for nucleus. So the prokaryotic cells do not have a distinct nucleus. They have a nucleus region, and the eukaryotic do. We as humans fall under the prokaryotic, under the eukaryotic. The prokaryotic cells you will discuss when you take microbiology. That's where bacteria and other more of the archaic organisms come in. Okay, so here's prokaryotic. So now that's a little example just to show you. So we are eukaryotic. It's a larger cell. It's more organized. It has more small structures called organelles in it. So there are several different features as we go along. Plants would also be eukaryotic. Here is just a schematic of a generalized cell that you can examine. Okay, when we look at cells, we look at the size. As you know, they're very small. The prokaryotic, as you can see, are much smaller. A micrometer is 10 to the minus sixth. 10 to the minus sixth. And the animal are 10 to 100. Okay, micromillimeters. All right, so we go here. We talk about the microscope. There are two kinds of scopes, the light microscope and the electron microscope. The light microscope uses white light as its source of illumination. The electron microscope uses something called electrons, as you may recall. Electrons are smaller than the light particles. One light particle is called a photon, P-H-O-T-O-N. The magnification and resolution, clarity, magnification and resolution is better with the electron microscope than the light microscope. However, anything being seen under the electron microscope must be dead, because a flow of electrons is tantamount to electricity. Let me say it again. What is electricity? A flow of electrons. So the organism must be dead. It has better magnification and resolution, but you cannot see organisms as they live, like you can with the light microscope. Okay. Magnification versus resolution. Magnification is the ratio of an object into its real size. So you pretty much, nobody has to, that's pretty common. Magnify. Resolution, which you call clarity, basically is this, is a measure of the clarity of an image. It is the minimum distance two points can be separated and these still be distinguished as 2 points. Because the closer you put 2 points together, in order to say these are 2 points, something must go between. Some illumination must go between. If a photon is bigger than an electron, then you can't put the 2 points together as close as you could with an electron. See, in other words, the sharp image. You heard of the high density televisions, high resolution. How close can you get these together and still say they are two distinct points? That's what we call resolution. So you can have a tremendous amount of magnification, but if you don't keep up with the resolution, when the big screen TVs came out, 20, 25 years ago, you could make a big image, but it was all blurred. So magnification and resolution. There are different kinds of light microscopes. You can resolve at about 0.2 micrometers and these are the different types of light microscopes. You'll talk a lot about that in microbiology. Here's some images you can just kind of glance at. The electron microscope can do a 0.2 nanometer versus micrometer. That's about 10 to the third smaller. So my point is that the electron microscope can resolve much better, but the organism must be dead. Okay. The reason I bring up the electron microscope is because in the text you will see pictures both from light microscope images and electron microscope. There are a couple of kinds of electron microscopes, the transmission type and the scanning. The transmission type passes through the specimen, so you see inside, like you'd be looking inside a cell. The scanning does not. To do the scanning, they coat the image with a metal. So when the electrons hit the image, they bounce off the surface to give a different image of the outside. Here's an example of a transmission electron microscope, and here's a scanning electron microscope. For example, here would be a scanning. These are cilia on top of a cell. So you see it, it looks like, like grass in a field. But you see on the outside. Here's a transmission, and it's going straight through. And you're seeing the interior of these particular structures. Okay? So what I want you to do is to really get down and study these cell components. These cell components. I want you to look in and kind of make you maybe even a little chart to talk about the various cell components. Okay. The first thing we come to when we're dealing with the cell components is the cell membrane, also called the plasma membrane. In earlier times, it used to be thought, oh, it's just the boundary, but now we know it does a lot of functions, a considerable amount of functions, the cell membrane. One of the big functions, obviously, it separates the intracellular fluids from the extracellular fluids. So here are some other functions of the cell membrane besides that. It provides shape to the cell. It has selective permeability. Permeable means to pass through. So it will let some things in and out, and some things it won't. And some things it lets in faster than others. It can also have receptors on the surface. It can also act for surface markers. Surface markers would be like flags to identify your cell from someone else's cell. It can also, some of the entities in the cell membrane can act in an enzyme function. The modern concept of the cell membrane is what we call the fluid mosaic model. The fluid mosaic model. Now, a mosaic pattern means there's just like you had a mosaic shirt, you would have several figurines on your shirt, several figures on your shirt. Fluid means it floats around. It consists of phospholipids, cholesterol, glycolipids, things we talked about in Chapter 2. Here would be an example to show you. The cell membrane, now I want you to read on this, has a phospholipid bilayer. Now these here, as I talked about in Chapter 2, these are phospholipids. The head of the phospholipid likes water. See, water and fluids have to come up to the cell membrane. If it didn't, but here's the key. Why have phospholipids? First of all, you want the cell to be in water, so it needs to have water touch it from the outside and inside. But you don't want water to freely be able to go through. Otherwise, you'd get dehydrated or things would occur. So what you do is, the phospholipid, remember it was amphipathic, has the hydrophilic section on the outside. So water would like it. But the hydrophobic on the inside. So it will allow water to get to the surface here, but not freely go through. Now it also has proteins. It also has cholesterol. All of these do a vital job. It has peripheral proteins just sitting on the surface, both the external surface and the internal. Then it has integral proteins that go straight through. Integral means important. That means if you slide it out, the membrane would start to kind of wither in that area. So the membrane has several different kinds of molecules, all have a certain amount of importance. We go back, a double bilayer, a phospholipid bilayer is what it consists of, with cholesterol, glycolipids, some proteins, and everything embedded within. Here again is showing that phospholipid bilayer that has been split to show you these proteins, to kind of give you an idea. An extracellular layer and an intracellular layer, and that in between. Okay. Here again I wanted to show you the phospholipid that you can read on. Hydrophilic heads, hydrophobic tails. That keeps that water from going straight through, but yet water can come on both sides. You can read by important aspect there. Here again is a phospholipid, just to refresh your memory. Here is the hydrophilic head because it's charged. Remember we said a charged group. Hydrophobic tails, which are fatty acids, they are not. Now these particular fatty acids are partially unsaturated. Now we always hear that animal fat is saturated, and that is true for the storage fat. But for structural fat in a cell membrane, it can be slightly unsaturated. Remember, a double bond means unsaturated. The reason for that, remember the cell membrane is fluid. So by having these tails crinkled, see, a double bond crinkles it, one phospholipid can't get too close to the other. Because remember, in a solid layer, when something's solid, things get close to one another. And you don't want that, so you want them spread apart. So these tails like that would be spread apart. Okay? So that's what we're looking at there. Proteins in the cell membrane. Two types. Integral proteins can either be transmembrane or partial transmembrane. Now, a transmembrane means it goes entirely through. A transmembrane means it goes entirely through. A partial means it goes most of the way through, but not necessarily all the way through. Okay? Then we have proteins that are just stuck to the surface. Now, those proteins stuck to the surface are not just sitting there by themselves, because otherwise they would get flicked off. So these particular peripheral proteins are bonded to the phospholipid, or to another protein type of thing there. So in other words, these proteins are bonded. If they're bonded to a lipid, they would be what? A lipoprotein. All right, so the peripheral proteins have functions too. So integral proteins and peripheral proteins. Here are the transmembrane proteins, just to show you. Here peripheral. So that one's bonded to this protein here. Okay. I'll take up that in a moment, again, talking on the proteins in the cell membrane.